Imagine you're working on a promising drug candidate. The synthesis looks clean, the biology is strong, timelines are tight. And then someone asks a deceptively simple question. How confident are we in the data? That single question is where analytical method development and validation truly begins. In pharmaceuticals, analytical methods are not just laboratory procedures. They are the backbone of decision-making, impacting batch release, stability claims, regulatory approvals, and ultimately, patient safety. Today, we'll walk through how modern analytical science has evolved, how regulatory thinking has matured, and how you can develop methods that are not just compliant, but robust, scalable, and future-ready. Let's start with the regulatory landscape. Not as a checklist, but as a philosophy. Earlier validation approaches were static. You developed a method, validated it once, locked it in, and hoped it survived real-world variability. Regulators quickly realized that this mindset was risky. That's why ICHQ2R2 and ICHQ14 represent a major shift. ICHQ2R2 modernizes validation expectations, allowing advanced analytical techniques and emphasizing documented validation protocols and lifecycle thinking. In parallel, ICHQ14 introduces the concept of the Analytical Target Profile, or ATP, a clear upfront definition of what the method must achieve. Instead of asking, "Does this method work?" we now ask, "Is this method fit for its intended purpose across its entire life cycle?" USP less than 1225 greater than complements this by categorizing methods based on risk and intended use, ensuring that validation effort is proportional, not excessive. This shift is subtle but powerful. Method development is often taught as a step-by-step process. In reality, it's far more iterative. Every decision you make, column chemistry, mobile phase pH, detection mode, feeds back into method robustness, sensitivity, and regulatory defensibility. Think of method development as building a bridge. You don't just check if it stands today, you ensure it withstands weather, traffic, and time. Most pharmaceutical methods begin with HPLC, and for good reason. In practice, sample preparation is often the most underestimated step. Poor cleanup leads to ghost peaks, column fouling, and irreproducible results. Filtration, solid phase extraction, or protein precipitation aren't optional, they're foundational. For column selection, starting with C18 reversed phase is standard, but experienced analysts know when to pivot. Phenyl phases improve aromatic selectivity. Helic becomes indispensable for highly polar compounds. Mobile phase optimization is where chemistry truly matters. Buffer selection, organic modifier choice, acetonitrile versus methanol, and precise pH control determine ionization, retention, and peak shape, especially for ionizable APIs. Gradient development isn't about speed alone. A 5 to 95% organic gradient gives you a wide lens early on, allowing you to balance resolution against runtime intelligently. Detection strategy should follow analyte chemistry, not convenience. UV and DAD work beautifully for chromophoric compounds, but ELSD or LCMS becomes essential for non-UV absorbing molecules. Gas chromatography demands a different mindset, precision through control. Column phase selection defines selectivity. Nonpolar phases work for hydrocarbons, while midpolar phases unlock separation for volatile polar analytes. Carrier gas choice is no longer trivial. Helium remains popular, but hydrogen offers speed advantages if safety controls are robust. Temperature programming is the real engine of GC performance. A controlled ramp ensures sharp peaks, even for high-boiling compounds. Injection parameters, split versus splitless, can make or break trace-level analysis. And for thermally labile compounds, derivatization is not a workaround, it's a necessity. LCMS combines chromatographic separation with molecular-level detection. And with that power comes responsibility. MS-compatible mobile phases using volatile buffers like formic acid or ammonium acetate are mandatory. Even trace non-volatile salts can compromise sensitivity. Ionization source selection, ESI or APCI, depends entirely on analyte polarity and matrix composition. Tuning capillary voltage, gas flows, and collision energy is not a one-time task, it's iterative. Matrix effects are the silent killer of LCMS methods. Post-column infusion studies reveal ion suppression that simple validation studies often miss. When suppression appears, improved sample cleanup, not software correction, is the real solution. Quantitation strategies rely on matrix-matched calibration curves, frequent system tuning, and vigilant monitoring of calibration drift. Validation isn't about ticking boxes, it's about confidence. Accuracy tells you whether you're measuring truth. Precision tells you whether you can do it repeatedly. Specificity ensures nothing else is lying to you. Linearity with correlation, coefficients above 0.995, reflects not just mathematical fit, but analytical discipline. LOD and LOQ are not theoretical numbers. They must be demonstrated under realistic conditions. Robustness testing is where experienced analysts shine. Small changes in pH, flow rate, or temperature reveal whether a method is resilient or fragile. System suitability is your daily insurance policy. Plate count, tailing factor, resolution, these aren't statistics. They're early warning signs. In pharmaceutical analysis, method selection depends on the question being answered. Routine assays often rely on robust HPLC methods, while impurity and stability studies demand higher sensitivity and resolution. For complex biological matrices, LC-MS/MS becomes essential due to its selectivity at low concentrations. Ultimately, the analytical technique must align with the analyte, matrix complexity, and regulatory expectations, because reliable data drives reliable decisions. Every lab faces issues. Poor peak shape often points to injection overload or column degradation. Ghost peaks tell stories of contamination or inadequate washing. Ion suppression usually whispers before it screams, if you're listening. The difference between junior and senior analysts isn't avoiding problems. It's recognizing patterns quickly and responding decisively. The future of analytical science is already here. We are moving from static validation to QBD-driven development, and now toward AI-enhanced analytics. ATP-driven methods allow accelerated optimization. Automation reduces variability. Lifecycle management replaces reactive troubleshooting with predictive control. But none of this works without people. Data literacy, analytical intuition, and regulatory fluency are becoming core skills, not optional extras.